Explore your knowledge with the Botnica.
Our new topic is DNA barcoding. It is a very important topic related to examinations, also asked in competitive examinations or useful in optional examinations. So what is DNA barcoding? So DNA barcoding, it is a method of species identification by using short section of the DNA. The short section of the DNA called as barcode. This is a short section of the DNA. This short section of the DNA are isolated from species, gene or genes. So simply DNA barcoding, it is a method of organism identification, or this is a method of identification of particular species. Uh, to done the identification, it is using the short section of the DNA of that unidentified organism. This DNA, the short section of the DNA are isolated from the gene of this unidentified organism. This short section is called as barcode. So now we have to make a roadmap in our mind that what is the steps to identify the organism or species by using barcode. Firstly, we have to take the section of the DNA from unidentified organism gene or from their genome. This section is known as barcode, and simply this barcode is used to identify the organism. So we have to know that about DNA barcode in detail. So as we see, that is DNA barcode, it is a short section of the DNA. So these short section are generated from the standard zone of genome of that unidentified organism. This short section, this region from where this short section get generated, this region is known as marker. So firstly, we know that about the marker. What is marker? Marker is standard zone of the genome from where the short section of DNA generated. So markers are the different for various species. In case of plant, this marker is MATK. In case of animals, this marker is CO1. And in case of fungus, the marker is ITS. This MTK, CO1 and this ITS, it is nothing but the markers used to markers from which barcode are generated, and this barcode used in DNA barcoding to identify the organisms. Now these short sections are stored in a library to use as we need or use in the future. Now this method of DNA barcoding is increasingly used to for the obtain taxonomic information of the about unidentified species, and it will make more easy to identify the unknown species by using the short section of the DNA instead of using a whole genome. There is no need to using the whole gene to study or to identify the particular species. There is need only short section of the DNA. DNA barcode are used in both cases in identification of eukaryote as well as prokaryotic organism. So finally, the steps of DNA barcoding. In the first step, we have to build a laboratory in which the short section of the DNA of known species are stored. Now, if we want to, in next step, there is a matching of unknown species section of the DNA with the known section which is stored in the laboratories. Then the identification get done. Finally, we have to see that there are some applications of the DNA barcoding. It is help in species identification, as we see. Next is to obtain taxonomic information. If you want to know about the taxonomic information about the particular species, then particular species gene, family, their genus, or you want any information related to this organism, Then you have to use the DNA barcoding. Next is easy method. It is an easy method of identifying organism. Why it is easy? Because it uses small section of the DNA instead of whole genome. Also useful in identifying alien species, and it is also useful in identifying the alien species, and also used in economics ecological survey to identify different species rapidly. So this topic of DNA barcoding get over. So thank you.